Pfizer's new COVID-19 drug, PAXLOVID, a very safe and effective pill that reduced the risk of hospitalization or death by 89%. 89%. Well, first, I'm going to explain everything you need to know about this drug, including the pharmacodynamics and the pharmacokinetics, basically how this drug works in the body and how it stops SARS-CoV-2 from replicating. Then after that, we'll go over the results from the interim analysis of the phase 2-3 clinical trial about this drug called PAXLOVID. What's going on, everyone? I'm James, founder of Health by James. I have a strong background in medicine, research, statistics, and patient care. I cover trending medical data and I break it down for you in easy to understand lectures. That way you can wrap your mind around the most current and important health trends that will ultimately end up impacting your life. Essentially, I'll keep you educated so you can make informed decisions about your health. But first, please like this video, subscribe to the channel, and hit that bell in the bottom right-hand corner to be notified when my new videos come out. Also, please help support my channel by clicking the link that says buy me a coffee below, or you can subscribe to my Patreon. All the links are in the description below. If you support my work, you'll receive exclusive content like more videos, my personal medical publications, educational content, and opinion pieces on the latest health trends. Anyway, let's get into this. To begin, I want to pull up this website real quick. You need to know about a couple of things here. So, you need to know about the history of this drug first. Now, if you look at the third paragraph here, it says PF7321332 was developed by modification of an earlier drug named lufeltravir. Now, let me open this real quick, 1 second. Now, look at the last sentence. It says, this drug was originally developed in response to the 2002-2003 SARS-CoV-1 outbreak. That was 19 years ago. So what I'm trying to tell you here is this PAXLOVID drug, under the different name lufeltravir, with a few differences, was already more or less developed. Importantly, originally, it had poor bioavailability, which is why it had to be given via IV. But now that's been changed. This PAXLOVID is a more bioavailable form or pill that can be taken at home instead of through IV at a hospital. Anyways, moving on, before we go any further, you really need to understand how this drug works or its mechanism of action in the body. So PAXLOVID is an antiviral drug that needs to be started within days of COVID-19 diagnosis. Also, it doesn't kill the virus. It only prevents further replication. That means it can't kill currently infected cells. It can only prevent future viral replication. Moving on, PAXLOVID is classified as a protease inhibitor. Protease inhibitors have a very interesting mechanism of action. They stop viral replication by stopping protease, in this case the coronavirus's proteases or cellular scissors, from breaking down large viral protein precursor into smaller, more usable proteins that enable the virus to continue replicating in our cells. Let me explain more. Normally, our cells produce usable sized proteins or workers that have specific functions. If they're initially not the correct size, our cells deal with that. One protein might be involved in building cell membranes. Another protein's job might be to control protein synthesis within the cell. So those are acceptable sized proteins our bodies can use. On the other hand, SARS-CoV-2 codes for many longer than usual sized proteins that are unusable to the human body. So what I'm trying to say, something has to break those unusually large viral produced proteins down so the virus can continue to multiply. So those cellular scissors that break down or cut big proteins are actually enzymes called proteases. And the drug we're talking about today is a protease inhibitor. In other words, it stops protease from doing its job. Now, the specific type of protease being stopped by this drug is the protease in the coronavirus called 3CL or MPRO, which is a chymotrypsin-like cysteine protease. Let me pull this visual up so you can understand, and we'll go over this again quickly. Okay, so when a virus like SARS-CoV-2 attaches to our ACE2 receptor and infects our cell, it then dumps genetic material like RNA into our cells. As you can see here, long story short, that viral RNA then links up with our human ribosomes to produce mainly viral polyproteins or big proteins. Well, after that point, SARS-CoV-2 can't continue to replicate in our cells using those big proteins. It needs those big proteins to then be broken into smaller ones. So those enzymes we just talked about, called proteases from the coronavirus, are supposed to come over and chop down those polyproteins or big proteins into smaller ones. So if you look here at this image I'm about to pull up in 1 second, if someone takes the drug PAXLOVID, the drug binds to the scissor portion of the protease so it can't cleave or cut the big proteins down into smaller ones. After that, future viral replication is stopped. Importantly, this 3CL or M-Pro protease isn't the only protease SARS-CoV-2 uses, but it's the main one that makes about 11 or so chops in the big proteins. So it's a big deal, and that's why scientists decided to target this particular protease of the SARS-CoV-2 virus. Okay, that is what this drug PAXLOVID does. Surprisingly though, PAXLOVID isn't one drug, it's really a combination of 2 drugs. It also contains another drug called ritonavir, which is an antiretroviral medication historically used to treat AIDS and HIV. Interestingly, ritonavir is also a protease inhibitor, but more importantly, ritonavir delays the exit of the previously mentioned molecule or drug, PAXLOVID, we were just talking about, from the body, so it can exert its antiviral effects on the body longer. Let me pull up this image so you can better understand. Let me make this a little bigger. Okay, now look at the left. That circle represents a hepatocyte or liver cell. When you take ritonavir or really many other drugs, eventually the drug makes its way to your liver. In the liver, you can see in the middle here, the body uses CYP enzymes, also known as cytochrome P450 enzymes, to break the drug down by doing different things like adding OH groups, etc. When you look here, the drug is broken down into metabolites, which are then excreted. However, ritonavir actually reduces the action of those CYP enzymes in the liver. Reducing CYP activity in the liver slows down drug metabolism and allows the drug to remain in your body for a longer period of time, so it works for a longer period of time. CYP enzymes do many important things like oxidize steroids, fatty acids, and xenobiotics, which are actually harmful foreign substances that can harm the body. So as you can see, you don't want these CYP enzymes suppressed for too long, as damage to the body can occur. Drugs like this that inhibit CYP enzymes have been shown to be hepatotoxic or harmful to the liver. It just depends on the dose and the length of time taken. I'm not saying the drug we're talking about today is harmful. I'm simply saying historically, we're careful with drugs that alter cytochrome P450 function. So that's how this drug works. Now, let's check out this study, or should I say Pfizer's interim analysis of their phase 2/3 clinical trial of their new drug PAXLOVID. This is on their website. Hold on, let me pull this up real quick. Now, remember, this has not been peer-reviewed yet. This is just a press release from Pfizer. Okay, so this was a randomized, double-blind, placebo-controlled trial of non-hospitalized, non-vaccinated adult patients with COVID-19 having risk factors like obesity, high blood pressure, that would leave them more susceptible to symptoms, hospitalization, and mortality caused by SARS-CoV-2. So that's good. The effectiveness of the drug in this study was based on hospitalization and severe illness, because believe it or not, other studies base endpoints or effectiveness on lab metrics or lab values. Well, you could prove a drug was effective by showing on paper that it worked, but if the definition of effectiveness doesn't take into account symptoms or even harm done to a patient, it might have been effective but also harmful. So what I'm saying is this is a good thing. Moving on. Okay, so participants were given the drug or placebo every 12 hours for 5 days, and looks like the treatment had to start within 3 days of symptoms, and then following that, all patients were monitored for a total of 28 days. Okay, now it looks like there were 774 total participants. If you look here, you see they were split up into 2 groups. The first group was the treatment group, which received the drug PAXLOVID. That group had 389 individuals. The other group was the placebo group. They received the sugar pill, essentially. Well, if you look here, hold on one sec. According to the data, in the treatment group, only 0.8% were hospitalized and 0 died. So essentially 3 people were hospitalized and 0 died. On the other hand, in the placebo group, a whopping 7% were hospitalized and 7 people died. So that's 27 people that were hospitalized and 7 total deaths. With all that said, if you look here, the drug showed an 89% reduction in risk of COVID-19 related to hospitalization or death. Now let's look at this. PAXLOVID demonstrated potent antiviral in vitro activity against circulating variants of concern, which is good. That just means in the lab on a petri dish or in a test tube, the drug was effective against the virus. That doesn't mean much though. We really need to see how this works against multiple variants in the human body, not just in the lab or in a petri dish. I'm sure you all know that. Now let's move on down here real quick. This trial contained people from North and South America, Europe, Africa, Asia, and USA, so it's diverse. And that's good because there are many studies on drugs, especially new drugs for COVID, that only look at certain populations of people. And that's bad because drugs in COVID affect more than just one population. They affect everyone. So the data needs to be based on a robust and diverse cohort or study group or population. Finally, what I didn't like, the amount of participants in this trial was way too small. There were only 774 people total. It's very difficult to extrapolate adverse events or side effects from such small numbers, so this data isn't really telling of what side effects would look like. However, there is some data regarding ritonavir as it is used in HIV patients. Also, there is a little data on the lufoltravir as it was developed years ago. However, all that data, it doesn't reflect safety and efficacy for use in the context of SARS-CoV-2 patients. Anyways, those are the facts, and if there's anything you'd like to learn about in the future, leave it in the comments section below, and I'll see you on the next one.